After DNA sequencing, Human Genome Project was completed in 2003, and after about 17 years, we still have not really gone very far. Okay, another type of vectors are called expression vectors. So you probably can guess, what expression vector would mean. Expression vector means those vectors that are used for cloning purpose also express protein, also produce protein. So whatever gene of interest you have inserted in a plasmid, that plasmid also is capable of producing protein from it. Insulin, for example, you will hear this example repeatedly from me because it is such an important and such an easy thing to understand. So when insulin gene is cloned, when inside the vector, and it is inserted inside a bacterium, then inside that bacterium, it is also able to produce protein. And that's what really we get from this process in case of insulin and many other proteins that are being produced in the same mechanism, like factor 4 proteins, which are used in kind of a blood clotting or other types of proteins as well. That may have more kind of a application. Most of those are actually used for clotting purposes. So those are all cloned in the same way, and our expression vectors are used to produce the proteins from them. So you kind of kill two birds with one stone. You're not able only to clone the gene, but you're also able to produce proteins from it. So those vectors look slightly different. They have this, kind of a marker, selectable marker that you can use them to select. They also have origin of replication for their duplication process, but they also have this structure here, this sequence here, which is a promoter sequence. So in promoter sequences, two important things. Pro is a promoter sequence, and ST is Shine-Dalgarno. Shine-Dalgarno sequence is found in case of prokaryotes, which is recognized by ribosomes for translation, for protein production. So the presence of this, followed by the gene of interest, gene is cloned in this site, which is shown here in blue color or purple, probably blue color. So once it is inserted here in the cloning site, promoter sequence and Shine-Dalgarno sequence help RNA polymerase to transcribe it. So the transcription will result, for example, here in the step 2, this gene cDNA is inserted here, and it is cloned inside a bacterium. At the time of transcription, messenger RNA will be produced, and then during translation, proteins are produced, and those proteins can be extracted by killing this bacteria. The bacteria are killed to isolate the proteins from them. In other cases, they have developed some systems where proteins are secreted. So those proteins are secreted outside the bacterium, so you do not have to kill bacterium in order to get those proteins, and bacteria can continue to grow and produce more proteins from them. Isn't that neat? So you don't even have to kill bacteria.